This lesson is about DNA replication, how the cell copies its DNA before it divides. By the end, you'll say where and when it happens, name the bonds that break, build a complementary strand, and explain why the result matters. Start with the question the exam always builds on: a cell is about to divide. Both new cells need the same genetic information. So where does the copying happen? Replication is DNA copying itself. But the cell does not do it at just any time. It happens in the nucleus, and it happens during interphase, before the cell divides. Why that moment? Because each new daughter cell must receive a complete set of genetic information. Copy first, then divide. If the DNA were not copied, one new cell would end up short. Now the Scratchpad asks you the same three things. Where does replication occur? When does it happen? And why must DNA be copied before cell division? Pause and try it. Pause, here is the full answer. Replication occurs in the nucleus. It happens during interphase, before cell division. And DNA must be copied so that each daughter cell receives a complete set of genetic information. Notice the three pieces: the place, the timing, and the reason. Now look at the diagram itself. You see the double helix, but the two strands have started to separate at one point. That Y shape is called a replication fork. The exam will ask you to label what is shown. The backbone is built from deoxyribose sugar and phosphate. Between the strands sit the nitrogenous bases. The order matters: first name the structure type, then decide the specific base. Labels 1 and 2 point to bases. So which bases? Use pairing to decide. In the Scratchpad, label the diagram the way the memo does. Label 1 is a nitrogenous base, and the specific base is adenine. Label 2 is also a nitrogenous base, and it is guanine. Label 3 is deoxyribose sugar. Label 4 is a phosphate group. 2 bases, 1 sugar, 1 phosphate, and you have read the figure completely. Before the strands can copy, they have to come apart. Which bonds give way? Between the paired bases sit weak hydrogen bonds. Those are the ones that break, and the two strands unzip or separate. Now the common error: the backbone does not break. The sugar-phosphate backbone is held by stronger bonds, so it stays intact. Weak hydrogen bonds break. Strands separate. Backbone whole. That is the memo's picture. The Scratchpad asks three things here. Which bonds break? Choose weak hydrogen bonds between complementary bases. What is the immediate result? The two DNA strands unzip and separate. Then correct the error: the backbone breaks. The backbone remains intact. Weak hydrogen bonds break, the strands unzip, and the backbone is left whole. Once the strands are apart, each one does the same job. Each original strand acts as a template, a pattern for building a new complementary strand. Two facts matter here. First, both original strands are templates, not just one. Second, to use a template you read one exposed base and pair it. Template G pairs with C, template A pairs with T. The rule never changes. 3 quick checks in the Scratchpad. A template is an original strand used as a pattern for a complementary strand. How many original strands act as templates? Both. And template base G pairs with C. That small set of facts carries a lot of exam marks. Building the new strand is mechanical, and that is a good thing. Work one base at a time across the template. A pairs with T, C pairs with G. When the rule is supplied, use it. When it is hidden, recall it. The pairing never changes. One more idea: the memo checks language. A single letter is a complementary base. A whole row is a complementary strand. The finished unit is a DNA molecule. Use the term the question asks for. Build both rounds in the Scratchpad. Round 1, template AGCTT with the rule supplied, the new strand is TCGA8. Round 2, no rule shown, template TCGAC, the new strand is AGCTG. Now the diagnosis. Template ACGTA, and someone proposed TGCTA, position 4 is wrong. It must be A, because T pairs with A. After replication you have two DNA molecules, and they are identical. That is the point of the process. Here is the detail the memo rewards: each final molecule contains one original strand and one newly formed strand. One old, one new, in both molecules. Why identical? Because the original strands act as templates, and complementary base pairing rebuilds the same sequence. A pairs with T again, G with C again. The code is copied exactly. The Scratchpad wants the result in words. Each final molecule contains one original strand and one newly formed strand. Two identical DNA molecules form. Why identical? Because each original strand acts as a template, and complementary base pairing produces two DNA molecules with the same genetic information. One old, one new, and the sequence is preserved. Replication is not an end in itself. It exists so cell division can happen properly. Follow the chain. Before division, DNA is copied and the genetic material doubles. During division, that material is shared between the daughter cells. And the result of mitosis. Daughter cells receive the same genetic information as the parent. Copy, share, identical result. That chain is worth 2 or 3 marks, so keep it in order. Complete the chain in the Scratchpad. Before division, DNA is copied and genetic material doubles. During division, the genetic material can be shared between the daughter cells. The result, daughter cells receive the same genetic information. Then write the explanation. Replication doubles the genetic material before division so it can be shared, and the daughter cells end up with the same genetic information. That is the full 2 to 3 mark answer. One confusion costs marks every year: replication is not transcription. Different jobs, different products. Replication copies the whole DNA molecule to make DNA. Transcription reads a gene to make a single strand of mRNA. 3 signals tell them apart. Replication uses both original strands as templates. Replication copies the entire double helix. And transcription uses uracil where DNA uses thymine. The products finish it. Replication makes 2 identical double-stranded DNA molecules. Transcription makes single-stranded mRNA. Classify each statement in the Scratchpad. Both original strands act as templates, replication. Uracil is used, transcription. The entire DNA molecule is copied, replication. Products, replication makes 2 identical double-stranded DNA molecules. Transcription makes single-stranded mRNA. 2 processes, 2 products, and now they stay separate in your mind. Every exam question on replication tests the same handful of ideas. So learn the chain once, and reuse it. Where and when, the nucleus, during interphase. What breaks, weak hydrogen bonds, and the strands unzip. The template role, each original strand acts as a template. The result, one original strand and one new strand in each molecule. The significance: daughter cells receive the same genetic information. When the question says explain, build that chain as cause and effect. When it asks for a visible reason, look at the diagram, not at background knowledge. Now the mastery check, 6 marks with no scaffolding. Replication occurs in the nucleus during interphase, before cell division. Weak hydrogen bonds break, and the two strands unzip or separate. Each original strand acts as a template for a new complementary strand. Each final molecule has one original and one new strand. Replication doubles the genetic material so it can be shared, and the daughter cells receive the same genetic information. And the visible reason: the whole molecule unwinds and new complementary strands form, with thymine visible on a new strand. 6 ideas, 6 marks. You can now explain DNA replication, where it happens, which bonds break, how each original strand acts as a template, and why the two molecules are identical. Open the Scratchpad again without the teaching, and write the full mechanism from start to finish. Copy, share, identical. That is replication.